So let's talk about this required practical doing chromatography. Now, chromatography is a technique which is good for separating mixtures. So you've got a substance, you want to know if it's a mixture. Chromatography can sometimes be the technique which it's helpful to use. Now, believe it or not, most people have already done chromatography. In primary school, you might have made something like that. You get some felt tips, you put it on some paper, you let some water go up it makes a pretty pattern. Well, believe it or not, this technique is chromatography. And what you created was a chromatogram. But what actually is it, and how do you use this idea to separate mixtures? Well, step one, you need to get yourself some chromatography paper. Now that is a special sort of paper, but I have seen this experiment done with filter paper. It tends to work almost as well. So you've got your chromatography paper, and job number one is you need to draw a pencil line on it, a couple of centimetres up from the bottom. And it is important that it's pencil for reasons that we will discuss in a little bit. You then get your substances that you are curious about. You want to know, are they mixtures? What is in those mixtures? And what you do is you put a blob of that chemical on the line, like so. You put a blob on there and you let it dry, and then maybe you get another one that you're interested in, and likewise you put another blob of that chemical on there, and then you let it dry. So now that you've done that, your next job is that you have to put the end of your chromatography paper into a solvent. Now, most commonly I've seen this done with water, but you can use other solvents like ethanol. Ethanol's good, but something that is a solvent, something that will dissolve the chemicals in your mixture. So you dip it in, and the solvent will start to rise up the paper, like so. And this is why it's really critical that the line gets drawn in pencil. When the solvent reaches this point, you don't want your pencil line to dissolve and start drifting upwards as well. Now, why is that? Well, you're going to need to use it to make some measurements. I'll show you in a minute. So let's say, for the sake of argument, that we let this run and run and run, and my brown ink, food dye, whatever, it slowly travels up and makes a dot just there. Now, what does that tell me about my brown dye, my brown ink? Well, it tells me that my brown ink is pure. How do I know? It's not separated out at all as it's gone up. It stayed as one continuous colour. Now, let's say that I look at my green ink, and let's say, like magic, it has separated out into a blob of blue ink and also a blob of yellow ink. A blob of yellow ink. Okay, that's not supposed to do that. I'm going to colour that one in by hand. Right, a blob of yellow ink. There we go. Perfect. So what does that tell me about my green ink? It tells me that my green ink is impure. It tells me that it's a mixture. And it tells me that it is a mixture of yellow and blue. Okay, so straight away we know something that we didn't know before. Brown ink is pure, green ink is a mixture. But it tells us something else as well. The fact that the brown ink has got a brown dot, or a brown spot rather, and that the green ink has got a yellow spot and a blue spot, tells us that these two inks don't share any chemicals in common, because the green separates into yellow and blue. The brown doesn't contain either. The brown just contains a brown pigment which you don't find in the green. So you can tell by looking at this chromatogram that these things are not they're not similar. They don't share chemicals in common. Now, there's other clever stuff that you can do with these chromatograms as well, and that is to do with something called an RF value. So what's an RF value when it's at home? Well, RF stands for retention factor. And essentially what it is a ratio of how fast a substance moves in chromatography. Okay, so how much is it, how is it retained? Retained, that's the word, retained. How fast a substance moves in chromatography is how much it's retained. So how do you calculate this then? Well, RF equals the distance moved by the substance divided by the distance moved by the solvent. Okay, best thing we can do here is to pick an example. So let's take my brown one as an example. Let's say I've let this run and run and run, and let's say that by the time that I decide to stop this experiment, the solvent has got as far as here. Okay, now what you can do is you can measure that distance. So how far, oops, that's not quite straight. How far is that distance there from that line to the solvent front? So what is this distance here? So let's say, for sake of argument, that it has moved, I don't know, 40 millimeters. Let's say that's what that distance there is. Okay. You can then actually, oh, right on the other side, that'd be a bit more helpful on the other side, right? 40 millimeters. You can then measure the distance from the center of your spot to your start line. So you're talking about this distance there. Let's pick a slightly more visible color. This distance there. And let's say, for sake of argument, that is 30 millimeters. Okay, I can now work out the RF value of this brown dot. So then that would be distance moved by substance would be 30 millimeters. The distance moved by the solvent would be 40 millimeters, which means overall it would come out as being 0.75. It's a ratio of how far that substance has moved. If it was 1, it would be right up at the top here with the solvent front. If it was 0, it would be right down here at the bottom, but it's not. It's 0.75. It's about 3/4 of the way up. Now the clever thing is that for this solvent and this this chemical, this one here, this brown chemical just here, the RF value will always be 0.75. And what you can do is you can look this up in a book. You can reference it, or I guess of a website or something. And you can look up that for this solvent, it got this RF value, and you can look it up in a book, and you can use it to identify what it is. See, it's clever like that. So, how does this actually work? What is actually going on here? Why do some chemicals travel faster than others? Okay, for that we're going to have to take a little bit of a closer look at what is going on with our chromatography paper. So imagine now that you are seeing the chromatography paper side on like so. Okay, and what you've got creeping up and through it is you have a solvent, and I think I used water as my example, so here it is. Okay, now these have got special terms in chromatography. The paper is referred to as being the stationary phase. And that's because it's not moving anywhere, it's not going anywhere, it's staying exactly where it is. On the other hand, the solvent, or the water in this particular case, is called the mobile phase. Okay, so what's going on here? Well, what you've got is you've got your pigment, your dye or whatever, and it is dissolved in the mobile phase, yeah? And what it's going to do is it's going to move from the mobile phase into the stationary phase, and it's going to move from the stationary phase back into the mobile phase. Okay, it's reversible like that. It's not a chemical reaction, but it does go forwards and backwards. And it is all to do with this idea of affinity. Does a chemical have affinity for the stationary phase? So does it prefer being part of the paper, or does it have affinity for the mobile phase? Does it prefer being part of the solvent? Well, the solvent's creeping up all the time. If it has an affinity for the solvent, as it's moving forwards and backwards and forwards and backwards between the mobile and stationary phase, because it prefers the solvent, because it prefers the mobile phase, it will move faster because it is hanging around in the liquid phase more often in the mobile phase. If, however, it has an affinity for the paper or the stationary phase, so it prefers to stay in the paper, so even as the solvent's rising up, it prefers to stay in the paper, well, that is going to mean that it travels more slowly. So the faster one is going to end up higher up the chromatography paper, and as a consequence it's going to have a bigger RF value, a larger RF value. And the slower one, well, as a consequence, it is going to be less high up the chromatography paper, and it's going to have a smaller RF value. Okay, so chromatography, great for seeing if something is pure or not, also good for identifying chemicals and seeing exactly what they are. Thanks.